years. So I'm going to welcome onto the stage our own Mike Kelly. He's going to do some groundwork, and then he's going to introduce our panelists. So this is the last science panel. Thank you, Mike.
Thank you.
Keep going.
Thanks a lot.
Thank you, guys.
Okay, guys, have a seat. Okay. Okay, good afternoon, everybody. I'm Michael Kelly. I'm part of the team at Cure Shane, part of the science team in there. And we've got a session here that's really talking about next generation technology. We're one of the few companies here that don't have a forward-looking statement. And it occurred to me whenever I was sitting in the back, I thought, oh, we should have had something. But our forward-looking statement is quite clear and quite simple. It really is about what the science is about here. You know, we're driven by gaps and weaknesses in the pipeline, and we're looking forward into how we can actually address those. And the way that we think about things is multiple years in advance. You know, we're looking at 2, 3, 4, 5 years down the line, because that's the pathway for drug discovery and drug development really to get a foothold. So our forward-looking statement is really technology like this that's set out to address some of the perceived weaknesses that you've heard about. We don't need to see me. One of the things that I've talked about constantly to people, and it's not a secret anymore, is that drug development is inherently unstable and really difficult. You know, the vast majority of projects that actually enter clinical development never get over the finish line or never be commercialized. And that number is about 90% of projects that are out there. And that's quite shocking to some people. You know, and they figure, you know, why on earth can drug development actually survive with that sort of attrition? Well, actually, pipeline attrition is a really important thing for us because it's where we really learn about the molecules we're putting forward or the targets that we're going, about the disease. And it allows us to fine-tune and modify because there is tremendous learnings in projects that actually don't make it or don't live up to their expectations. And that's always been the history of drug development. It's like 2 steps forward, 1 step back. And so all those gaps that you see in pipelines or weaknesses or failures of program, they really drive an enormous amount of innovation. And there's what you're going to hear about today, you know, from the panel that we've got here. This innovation is driven by perceived failures or weaknesses or drugs that don't live up to what we intended them to do. And there's just the nature of drug development per se. I can give you a few examples, you know, of things that we've got today that we didn't have a decade or 2 decades ago. And a great one actually is where we are with COVID. You know, the way that we were able to intercept that there with technology that seemed to come out from left field. Well, you know, the delivery particles, lipid nanoparticles have been around for a while, and the RNA technology had been around for a few years, but it was the inception of all that that allowed us really to jump in and treat that disease very, very quickly, where historically it would have taken years to build up vaccines. And there's other great examples in the antiviral field, you know, from hepatitis to AIDS, that we can do today with oral drugs that we couldn't do in the past because of failures and learnings and better understandings. I think for me, for this community, you know, I'd take you back to yesterday's session with the exon skipping drugs, because they're the first approved drugs were most welcome. You know, we clearly recognized that we could get a drug approved in order to treat this disease, but we knew instantly that there was limitations in those. And that drove a whole swathe of technology that got us to work the story that you heard yesterday. You know, there are a whole bunch of companies from Sarepta themselves, all the way through to PepGen, Avidity, Dyne, and others. They started to bring in different technology, recognizing the weaknesses that exist with those exon skipping drugs, targeting muscle with real fidelity, being able to get these oligonucleotides inside cells to enhance exon skipping. And the results that we've seen today, you know, they really, I think, hold tremendous promise and potential, you know, for the whole changing landscape of exon skipping drugs over the next few years. And that's what this is. You know, this is that turnaround of knowledge and information based on not meeting expectations. We've just come out of a panel run by Jeff, you know, really recognizing, you know, what we're doing right now as we're developing these gene therapy drugs. And like the few comments that I would make in there really to set the stage for what we're going to hear is one around, there is a tremendous amount of anticipation and excitement within the community for what these drugs can do. Like, we know they're more than 10 years in the making to get to this point. And the data that's been generated and presented so far really talks to a high amount of microdystrophin in muscle, correctly assembled at the muscle membrane. And the results of that there really are seen in time function tests, strength improvement, the North Star scores in these boys. And like I've no doubts, we've no doubts that this is going to have an enormous impact. I should have been on the panel actually because Jeff asked the question, but they are going to make an enormous impact as we go forward. But in keeping with the way I started this, we recognize even now as those first drugs are not approved, they're marching forward towards approval quite quickly. There's gaps and weaknesses that exist in there. And I've put a few of them on this list right now. And it came up in topic in the last discussion. You know, it really surrounds the fact that we know that not all patients are going to be treated. We know part of the issue in there is neutralizing antibodies and trying to deal with that. And we don't have an exact handle on the number of patients that would be excluded, but it could be north of 30%, it could be north of 50%, it could even be higher than that again, because AAVs are prevalent in society and people have been exposed to them. And we need to figure a way to deal with that. And that's part of the way these lessons that we learned is asking questions that technology is going to drive itself into. That neutralizing antibody leads itself on to redosing, and we hear that constantly from the community as we're starting to learn from the clinical trials about the benefit-risk ratio that we're seeing for these drugs prior to approval. We know that we're going to need redosing at some point. My sense is that as we dose younger and younger patients, the need for redosing is going to become more acute rather than what we might see with dosing an older individual, and that there's something that we have to deal with as well. The other one that constantly comes up is the size of the mini gene that's put in there. Now, it's clearly been designed with the brightest of minds in order to be functional in human muscle. But all the constraints around that design have been brought about because of the amount of space that we've got in the capsid to deliver it. And we clearly, if we didn't have that limitation, we would want to go on and design a transgene that was much larger, one that we see in a Becker muscular dystrophy patient, one that's associated with an asymptomatic disease, or even full-length dystrophin. And you're going to hear, you know, in some of these technologies, that's exactly what they're trying to amend and what they're trying to do. And then the last thing really is delivery. Well, it's not the last, it's the last I put on this slide, but it really is delivery. You know, these capsids, AAVs, don't have fantastic tropism for muscle. It was touched upon by Jeff that the design of new capsids that nature never intended is going to be an important part of going forward with AAV-mediated delivery. But we want to get outside of that. We want to get real tropism for muscle. And that there becomes critically important because it allows us to reduce dose or reduce toxicity. And that there's an important component of what you're going to hear from this panel as well. So the way that I've brought this, to, at least the way I've set this up, is to show that there is an enormous amount of learnings that we got whenever we done exon skipping drugs, and that's advanced and you witnessed that. I think the same is true today with where we are with gene therapy. And we're thinking way ahead, you know, of trying to hit these problems off, you know, in the future over the next few years. These technologies that you'll hear about, you know, from each of these companies are really designed to take us over the next few years to answer all these questions. And we've got 3 individual companies that we're going to introduce. The first is Chameleon, and we have Janine Winslow, their CEO. Chameleon's got a very interesting technology where they're shielding a virus from immune surveillance that's really there to address the redosing issue or to address the neutralizing antibody issue. The second is Aaron Brubaker, the Chief Operating Officer at Code. And Code has stepped back and taken a totally different approach that's non-viral. And because it's non-viral, they're using a targeting modality towards muscle and increasing that from tropism points of view. And then they're not restricted to size, so they're looking at much larger genes. And the final one is Joe McIntosh, the Chief Medical Officer at Geneo Bio. And again, using a somewhat similar approach, but an antibody that's specific for muscle and then can carry a nucleic acid carrier into the cell, which could be DNA or RNA. So with that, you didn't want to hear from me. I just wanted to set this up. We got 3 great presenters. Janine's going to go first, and I'll sit back and let these guys talk. There you go.
Okay. Thank you very much for the introduction. And I'll introduce myself first. I'm Janine Winslow. I'm the founder and CEO of Chameleon Biosciences. I founded Chameleon about 5 years ago. My dream then was to be able to treat all patients with gene therapy, and I was worried about durability even back then. So my goal was to come up with a way to improve upon current gene therapy technologies. I want to thank the organizers of the conference and thank you all for coming to listen to my talk. And I'll start very briefly by acknowledging the scientists that have developed the data that I'm going to show you. I will show you a little data. We're a very early stage company, just 11 people. They've worked really, really hard to get to where we are now. So, as I mentioned, I started Chameleon because I didn't want to exclude any patients. I didn't want to exclude the patients that were previously exposed to AAV and may have had those pre-existing neutralizing antibodies. And as it turns out, since we founded the company, our technology has other benefits. We have better potency per dose with our vectors compared to the current technology. That will help us with the cost of manufacturing. We anticipate ours will be lower. There's also, has been described in earlier talks, some of these severe adverse events in the clinic. We also are going to be able to address some of the causes of that because we're reducing the immune response to AAV-based gene therapies. That and our increased potency will allow us to provide multiple doses. And so instead of delivering one massive bolus, if you will, the one and done paradigm, we want to break that down into a few doses that are smaller and never reach those very high, more risky doses that we've had to use in the clinic so far. Lastly, I wanted to be able to use gene therapy to treat more kinds of diseases, not just rare genetic diseases, but rare genetic diseases affecting children whose tissues are still growing. They would need repeat dosing because this type of gene therapy gets diluted as cells divide and tissues grow. So that was our goal. And you see on these slides a representation of what our technology is. It's basically just like what's being used currently on the market and in the clinic. We've got an extra layer surrounding the particle that has been engineered to contain two really potent immune suppressor molecules. These are molecules that have been identified in immuno-oncology. Tumors use them to evade immune detection and immune killing. And so we're borrowing that knowledge and applying it to gene therapy. The way that we work and the reason we think we're working so well, which we are in animals, is because we're casting a broad net over the entire adaptive immune response and reducing activation in multiple stages with multiple cell types. Importantly, with repeat dosing, you have to target the memory B and T cells are the biggest components. Those are what we have to control to repeat dose gene therapies, and our technology seems to do that pretty well. I won't spend a lot of time here. I just want to say that our technology is literally built on the knowledge gained from other gene therapies that are in the clinic. So we've got a well-established roadmap, both for CMC, which is manufacturing, as well as regulatory approval. So I have a bit of data, and for those of you who are not used to looking at data, this basically tells us that in mice, we can deliver a second dose of our technology and it will be just as effective as the first dose. So that's what doesn't happen with existing technologies, and it's what our technology was designed to do. We were thrilled with this, and now we've gone on to test our technology in larger animals where after 2 doses it seems very safe, and very preliminary data seems to indicate it's working well. We were really excited to find with our technology that we do reduce the immune response, different components of it, as I said, B and T cells, and most excitedly, we seem to be reducing inflammation. We're working with an AAV8 factor 9 clotting factor model. For various reasons, that was the fastest way for us to get proof of concept. We would now like to apply our technology to targeting muscle diseases and specifically Duchenne muscular dystrophy. We make our product just like current AAVs, and the benefit is we do get to leverage lessons learned. both about immunology in the clinical trial results as well as about manufacturing. And in both of those areas, we've had a lot to learn over the last 6 or 8 years. So we have increased potency, at least with this model in mice. We get about a 10-fold increase after 2 doses with our technology. We maintain tissue specificity. That means we use the same capsids that everyone else is using to target the same tissue types. That's huge because we don't have to reinvent the wheel. We can leverage what others have done. And we think that we can use our technology for engineered capsids, which I'm really excited about because then we should be able to get the best of both worlds, the benefit from the engineered capsids and the benefit from our immune suppressive extra layer. We will not be excluding patients in our first clinical trial based on neutralizing antibody data. This is what we're planning, and that was our goal. We have significant resistance to neutralizing antibodies, and I'm very excited to be able to take the field in that direction. And then, as I said, we have been able to repeat dose safely and very effectively, now just for 2 doses, but we're working our way up to 3 in animals. Very briefly, our goal, as I said earlier, is not ever to have to deliver those very high doses that cause the adverse events. We'll stay in the safer dose range, if you will, where we can administer multiple doses to get sort of a cumulative gene delivery, if you will, to get maximal clinical efficacy. So I'll end there. I think I'm out of time, and I'll pass it on to the next speaker or to Michael. I'm not sure. I think it's you.
Erin.
Okay, Erin. Thank you.
Hi, everyone. I'm Erin Brubaker. I'm the Chief Operating Officer at Code Biotherapeutics. And with Code, our mission is to treat the untreatable and cure the incurable. And as you heard Mike describe, our approach is completely different than anything you've heard, I think, in the last day or so. We really admire the work that's been done in gene therapy, in RNA delivery, as well as in gene editing. But we feel like there's still an unmet need out there in terms of delivering these genetic medicines. And that's where we see our technology coming in. So I am a non-scientist, so you're going to hear a non-scientific explanation in this slide and the next slide. I welcome you to go to our website. There's papers that you could read, and certainly love to talk more about it. But at a very high level, where we're different is that we are a non-viral 3NA delivery platform. So essentially, what that is a synthetic DNA that's formulated using chemistry and is able to deliver genetic medicines at a higher targeting specificity, which we believe will lead to lower doses being used in patients, which could be potential for a better tolerability program, or profile rather. We also believe we could be capable of delivering larger payloads because we're not constrained to AAV since we are a non-viral delivery. We, as you'll hear, what we're planning on doing for a DMD, we're looking at larger gene delivery. We also are non-immunogenic, so we've tested that, and I'll explain a little bit more in terms of our immunogenicity profile on our 3DNA. We're not seeing any. We haven't been in humans yet, but we're not seeing any immune response to the 3DNA scaffold. So we believe that could also allow us to do redosing, which would be very important. And then the last is the scalability of our manufacturing. So this is how our platform works, essentially. And the scientists that I work with would be mortified, but I think of it almost like Legos in a way, because you're basically combining different pieces. So you're taking you're building a scaffold of synthetic DNA through chemistry, and we've found a way to eliminate some of the nucleotides that are known to cause immune responses in humans, and we've taken those out of this process. Then we tack on different targeting moieties. So we can really specify how we're going to, the diseases that we're trying to treat by using different moieties to target the scaffold to get into the body, as well as to add different promoters and to deliver different payloads. So what this looks like when you're seeing it come into the cell is basically this 3DNA scaffold, which would be delivered through an IV infusion. would have a targeting moiety on the front of it, which would bind to the cell, and then would enter into the cell. And then the way that the 3DNA scaffold works is that when it hits the lower pH, it essentially dissolves in your body. And so it's able to have the endosomal escape, so the actual promoter and the payload are traveling to where they need to get to in order to develop a therapeutic protein. We've conducted years of studies on this from a preclinical standpoint, which are animal studies, and we've developed a lot of different data in different targeting tissues. So while we're focusing specifically for muscle in our DMD program, we've also generated data in multiple organ systems. So our pipeline today looks like this. We've got a Duchenne's muscular dystrophy program, which I'll talk a little bit more about in the next slide. We also have a few programs in type 1 diabetes, and then we've got some business development partnerships with big pharmaceutical companies, similar to what Deborah had talked about yesterday in the opening session, really making sure to leverage all of the ways that we can accelerate the science. So our DMD program specifically, we're looking at a few different cell line models using DMD patient skeletal muscles, which is why it's so important to be able to partner with Cure Duchenne and the LINK program, as well as MDX mouse models. We're using specific muscle targeting and promoters, and then we're swinging for the fences and trying to deliver the full dystrophin gene. We do have a few backup programs in case that doesn't work, but that is our fervent hope, is that we're able to deliver the whole gene. These are my colleagues on the executive team who couldn't be here today, but they are very passionate about delivering new and innovative medicines on a variety of different diseases, including Duchenne's. And we have a whole team of scientists behind us in the Philadelphia area, which is really turning into the home of cell and gene therapy across the US. We're also supported by an exceptional number of scientific advisors, Mike being one of them, and as well as a few other names on here that you might recognize, either through the Duchenne's community or through drug development in other companies at Spark, and having experience at the FDA and as well as Penn. And we are a small private company right now. So we raised seed funding last year, around $10 million. We're still using that today to really continue to advance the pipeline. Some of our investors, fortunately, we were able to partner with Cure Duchenne Ventures here, and they've been fantastic about providing both scientific advice as well as seed investments, as Deborah mentioned in the opening session. We've also had investors from UPMC in Pittsburgh, NEA, the Juvenile Diabetes Research Fund, similar to Cure Duchenne Ventures, 4Bio Capital, which is a VC fund that specializes in genetic medicines, and Takeda Ventures. And Takeda has also come back to us and created a partnership on developing drugs in the liver and CNS fields as well. And we're in the midst of raising our Series A now, so we can continue that pipeline progress. So I would just say thank you so much for the opportunity to tell you guys a little bit about Code Bio. Thank you for sharing your stories and your insights and your samples with drug developers, because it's so critical to what we do, and you're very inspirational, all of us. So thank you so much.
Good afternoon, everyone. My name is Joe McIntosh, and I'm the Chief Medical Officer at Geneo Bio. Just to give you a little bit of my background, I'm essentially a drug developer by pedigree. I've been in drug development for 20 years, in rare disease for 12, and I worked in Duchenne for 7 years. So it's a pleasure to be here and to present what we're doing, the exciting work that we're doing at Geneo Bio. All right, here we go. So Geneo Bio is developing a first-in-class antibody, monoclonal antibody that can deliver a gene therapy. So what is that? What is that? And we call it the GMAP platform. So essentially, it's a non-viral, cell-penetrating antibody that binds to a genetic payload and actually delivers it to cells. And currently, we've got robust data, preclinical data in animals that shows that we have targeted delivery of a broad range of genetic payloads. So that includes both RNA, it also includes DNA. We've also got some very exciting animal data, which I'll take you through. And what this antibody does, what it does quite uniquely, is it actually delivers these genetic payloads to two types of tissue: tumor cells and then skeletal muscle cells. So strategically, where the company is focusing, we're focusing on oncology as well as rare skeletal muscle disease. So what differentiates our particular antibody, our GMAb technology, from the existing gene therapy platforms that are available? And so this slide really breaks it down into 5 unique assets or aspects of the technology that differentiates it. So first of all, as I originally said, the antibody binds to these nucleic acids, to these genetic payloads. It binds to it in a non-covalent way, so we don't need to chemically link the antibody to the genetic material. So that gives us a lot of flexibility in terms of what we can actually do, what gene therapies we can develop. In addition, there's targeted cell uptake. So the antibody targets muscle cells and oncology cells, and it gets into the cell through a particular type of transporter called ENT2, which I'm going to talk to shortly. I'll explain some of the biology and how this antibody gets into the cell. Now, one of the unique traits of this antibody is normally when antibodies are taken up into a cell, it gets trapped into a little organ in the cells called an endosome. The problem with the endosome, it often degrades the genetic material that these antibodies are attached to. And because of the unique way this antibody gets into the cell, it avoids the endosome. So we believe that will really differentiate the antibody to deliver a much better genetic payload to a cell. Then something that's been spoken a lot about at this conference is the need and the ability to repeat dosing. And that's a very important differentiating feature. I think we're all now trying to develop technologies so we can give additional doses. And that will mitigate the risk of giving really high doses. We can give repeat doses and hopefully maintain and improve on the benefits that these genetic materials give to patients. Another very, very important feature is a safety profile. So obviously safety is on everyone's mind. And the nice thing about an antibody, antibodies have been in development for more than 20 years now. So we really understand the safety profile of antibodies, and therefore we feel that's a real benefit. So we have that advantage. And then last but not least, gene therapies are very expensive to manufacture. In particular, viral gene therapies, they're very costly and very complex. The good thing about this is it's an antibody. It's much easier to manufacture, much more affordable. Okay, so now I'm going to explain the biology of this particular. How does it get into the cell, and what does it actually do? So, as I originally said, this antibody, which originally was discovered in lupus mice, binds to a variety of genetic payloads. And so when it binds to that genetic payload, it does a couple of things. It actually protects the DNA. So if you put DNA or an RNA into a human being, the body will break it up. The enzymes in the bodies will break it up. By the antibody binding to the nucleic acid, the genetic material, it protects it from that breakdown. So that's very important because then it can be delivered to cells. Secondly, it then helps the genetic material get into cells, muscle cells and cancer cells. And it does that through a particular transporter called ENT2. So what ENT2 does is essentially it pulls the nucleic acids into cells and into nucleic acids, and then it delivers those nucleic acids and they express, so they become functional. So this is just a little bit of data which shows that a variety of different payloads can be taken up. And the colors you see are the genetic material getting into cells. And so we can deliver large genetic materials and we can deliver small genetic materials. So this gives us a lot of optionality. Now this is very important. This is showing you the levels of expression of the ENT2 receptor, which is the receptor that this antibody gets into the cell. And what you can see there, and those are RNA levels, expression levels, it's very high in muscle. So muscle is perfect for our technology to get into cells. We're looking at a variety of strategies. We can look at protein replacement, exon skipping, gene editing, knockdown. Duchenne is true to our hearts and is an indication that we're really looking to pursue. And we're in the process of gathering more and more data to pursue that. So this is just a little bit of data, my last 2 slides before I finish off. So this is data where we took human muscle cells and we looked at getting a skipping agent into it. And what you can see if you look at the slide on the right there, you see we gave just the skipping agent alone, and then we gave it with the complex, our antibody. And what you see by that red, because it's been labeled red so you can see it, the antibody is pulling that genetic payload into the cell. So it's telling us that the antibody is helping get this genetic payload into skeletal muscles. That's essentially in a lab. And that it's expressed so that you get it in and then it's working. The next experiment we did off this was to look at in a mouse, can we actually get a genetic material expressed in a mouse? And so we used something called luciferase, which is a protein that's luminescent, so you can actually see it in the mouse. And we injected that, the RNA from that, with our antibody and into the muscle. And what you see is that the antibody, you got the mRNA into the cell, then it would convert it to the luciferase protein, which then fluoresces, and you can see up to 5 days the genetic material is fluorescing. So it shows we can get genetic material in there and that it gets expressed, so that the protein gets produced. So in conclusion, we're very excited about this technology. We think it's highly differentiated. It's an antibody that penetrates cells and can deliver nucleic acids to both skeletal and oncology cells, and we plan to develop GMAB in both of those. And our next experiment is really to go into the MDX mouse and to look at data, and hopefully in the coming months we'll have some more data to share with the community. Thank you, everyone.
Thank you, all 3 of you. That was fantastic. It's a bit of a tour de force going through 3 different technologies and platforms. But the way that I described it at the very beginning was, we're looking forward to the future, of where the gaps are, and it's really guiding the way that we're thinking about this. And we've certainly talked about this to parents, as we're trying to get them to understand the pipeline and some of the limitations and how we're dealing with this. And the questions that always come up first with me are, when? What the sense of a timeline whenever they may see pivotal data, the sense of a timeline when these things are moving forward into the clinic. Janine, do you want to take a dig at that to begin with and give us a sense of where you are today and where you may be in 12 months?
Sure. I like to say we'll begin our sprint to the clinic in we're planning January of 2023. We're waiting to raise money, basically, to get into the clinic. It's expensive. And the manufacturing takes a predefined amount of time. It takes 18 to 24 months to really do justice to the scaling up and production development that you need to do to produce clinical material. And then there's all that has to go on in preparation for the clinical trial itself. So we're thinking late 2024, early 2025 would be when we would get clinical data, and I wish it was sooner. We're going as fast as we can.
Yeah. And Erin, I would change the question just slightly for you. I know that it's unfair to really think about clinical setting whenever the company is so young and generating early data, but I do know that there's key and critical experiments that are going on right now, specifically in Duchenne. And if you give, the community a little bit of a sense of when you might expect to see those and what that would mean to forward-thinking plans.
Sure. So we do have studies that are some that are ongoing right now, more that are planned for the rest of this year, and some more for next year. And very much like Janine, we're rapidly trying to get to a place where we can have a lead candidate that we call them, which is essentially what you're hopeful that is the medicine that you'll eventually be able to take to patients. So we're rapidly trying to understand what that lead candidate should be that's optimized, and make sure that we've got the manufacturing behind that in order to file an investigational new drug application with the FDA. So we'd love to come back and present data when we have it. Unfortunately, we are early too.
Yeah, and it's no secret. I think it was on the slide that we've been working together and sharing thoughts and providing KOLs into this work. But I would say to the community, there's a number of different shots on goal with this technology, and we're going to work our way through all of this. And we're leaving no stone unturned, essentially, in order to get success out of this.
Exactly. Yes.
Yeah, thank you. And Joe, one of the things I would ask of you is that the technology is quite flexible in that you've got the luxury of binding nucleic acids, and so you can go down an RNA pathway or a DNA pathway? Where's the pivotal work that's going to make you decide what you want to do as far as this is concerned?
I mean, that's a great question, Mike. I think for us, it's just getting in vivo proof of concept, so in animals for musculoskeletal. So, we are going to do an MDX mouse study, and that's going to be critical. Our strategy is, bigger payloads are a little more complex to get into cells. They're very negatively charged, and it's a lot more, you need a lot more antibody to get it in. So I think our initial strategy is to focus on smaller payloads and then move to bigger payloads in an incremental way. So that's certainly how we're thinking about it now.
Yeah, thank you. Look, I'd say for the latter two here, we've been toying with various ideas and strategies, and clearly, the ultimate goal is to get full-length dystrophin in there, and there's a capability of doing that.
Right.
But we recognize, it's an enormous lift for any technology in order to do that and to get it into all muscle, cardiac, as well as skeletal and diaphragm. So there's a number of things that one's looking at in here of recognizing that we want larger transcripts, but transcripts that nature has designed and recognized in Becker muscular dystrophy patients. And so there's a range of opportunities in here, a range of shots on goal is the way that I would put it. So the community should be really, anticipating a lot of data that's going to come out of this that's really going to guide our thinking over the next couple of years. Janine, back to you. One of the things that constantly comes up as I meet with families and parents is the data that we don't have, and that is the duration of the effect of gene therapy. There's nobody can give us a definitive answer right now, and that's not the question. I wouldn't dare put you on the spot. But, we're going to learn about this over a number of years. And as I mentioned in my introduction, I think as we dose younger and younger patients, which is something that we really have to do, there will be a need earlier rather than later for redosing. Do you anticipate that there with your technology and your thinking about a patient that has got neutralizing antibodies to begin with who would be excluded from AAV gene therapy is someone you would look at, or to redose an individual years past where they've received an AAV-mediated gene therapy? Can you share some of your thinking in there and how you're maybe even anticipating what a clinical study would look like?
Oh, sure, yeah, because it's actually really exciting, or I'm very excited about it. We, as I said, our proof of concept has been in Hem B. Well, why am I talking about hemophilia in a Duchenne conference? Because that particular disease was the fastest way for us to show proof of concept, and it was a way to show it with many, many years of clinical data on the immune response to the gene therapy. So it was a way for us to leapfrog. Now we're ready to take on a slightly bigger challenge, which is DMD. And in terms of the clinical trial, what we're doing in hemophilia will really pave the way for all of our subsequent programs. So, and it's actually quite exciting. We're planning a 2-dose minimum. So our first clinical trial, we're planning not to exclude any patients based on neutralizing antibody titer. And then each patient will be given a minimum of 2 doses. And so that's not been done before, and I'm really excited about it. And the reason that we think that we can do this safely, and I should preface all this by saying, we've spoken to leaders in the gene therapy field, and that's how we've come up with this clinical plan. We will be going to the regulatory, the FDA and the EMA agencies to get approval from them. So, we think we've got good scientific reason that it would be safe and efficacious to do these 2 doses. So we'll be treating patients with 2 doses within a very limited time frame. That sets the stage, though, for us to be able to come back years later and deliver an additional dose in case a particular patient starts to lose clinical efficacy. The other really exciting aspect of the idea of multiple doses that I think will help the field of gene therapy is this idea that each patient responds differently to gene therapy. Each patient's immune response is different, and that is going to affect how much of the therapeutic protein they make. And a classic example of that is, again, in hemophilia, where with the same dose, one patient made 2% of normal of the therapeutic protein, another patient made 200%. So by going using multiple doses, we can use the first dose to see how each patient's going to respond. And then if we need to, we can plan for more doses. Whereas patients that respond very well, we don't need to plan to provide more than one dose. And this is all, the reason we can do this is because of our immune suppressive strategy. That's what's allowing us to consider this really alternative clinical trial design. And so I think it's pretty exciting.
Yeah, I asked because we talked about it last night with a beer,
it's really
a
exciting.
beer in our hands.
Yeah.
As I go down the road, you know, it's a similar sort of question, but in a different way. You know, these technologies are not restricted because, like, non-viral technology by its definition is not influenced by that. And one of the things I would add, as part of the question, the half-life of dystrophin, as it's correctly assembled in muscle, is in the order of months. You know, so it really feeds into leveraging this type of technology. Right. It's not something that you're going to think of as a weekly or a monthly injection. It could be much longer. Right. So have you comments on the way that you're thinking about that from a target product profile?
Right. Yeah, I think I like the idea of catering your dose to the patient's needs. I think what we have seen, you know, specifically in the viral gene therapy, is a lot of variability, and I think the ability to redose. So if you can measure the protein, then you can dose in accordance to the protein. So with our technology, we imagine we'd need to dose initially to get a good amount of genetic material into the cells, and then, you know, I think infrequently based on need. So I think that's how we're viewing it now. But I imagine with this antibody we'd be dosing indefinitely, because I think you do need that continual genetic payload for growing kids as they enlarge. It will depend on the indication. You know, in Duchenne's, that, you know, over a period of time things will wane, so you will need that repeat dose. But we would load and then maintain would be the strategy we're thinking about.
Perfect.
Just to add to that, I would say that for us, we're looking at scientific advice as to the best way to approach it. I think the market is going to change, and I call it a market, but the disease area is going to change as we see these new medicines coming out and patients taking them. And so I don't know that, and I don't feel confident to comment exactly what we would need to do from a redosing standpoint right now. I think the other piece that we would need to do is get insights from the parents and the patients and the physicians that are treating you to really understand what's going to be needed in the future. And I would also say from a payer perspective, I mean, the world we live in is also from a payer perspective. We're going to have to understand what insurance companies are going to need to see. So, you know, when we look at the clinical trial development design, it's going to be a totality of really what patients and parents and the physicians that are treating them, the advisors and the payers are going to look for.
Super. We got 1 minute, and I want to spend it on what's always the elephant in the room in drug development, which is safety. You know, we're always constantly surprised. You can't predict it quite often, and talks can be idiosyncratic at best. But I think from the SAEs that were described at the end that we're starting to recognize as part of the risk-benefit that's coming out from. Could you give a comment on where you think you would stand, and that's on the technological approaches that you've got and how this might present, or anything else that, you know, concerns you?
Sure. I mean, one of the things that we're excited about with our platform is it's an antibody. I mean, antibodies have very defined safety. We've just got a humanized version, so that'll help specifically with the safety antibodies. So I think with antibodies, they've been around for 20 years, so we have a lot. We don't have to worry about immunogenicity, which I think is the biggest challenge. So we're pretty excited about that, and we think it'll have a much more defined and manageable safety profile.
Yep.
For us, it's a synthetic DNA that we've created. So, and we've stripped out what we believe are the nucleotides that are actually going to cause immunogenicity. We're not seeing it at all as a result of the scaffold in our animal models, and we'll let you know what happens when we get to patients.
Last word, Janine?
You know, we are addressing the safety by starting low, starting with a single dose, making sure that any given patient is not going to respond in an unsafe or adverse way. And then, as you know, we always have the opportunity to introduce more immune suppressive drugs if we need it. Right now, we don't need it with our vector, but I think the idea of starting low and then adding, because our durability should be months to years. We can deliver multiple doses in over a month or a 2-month time frame, and hopefully that will last patients for several years before needing a reboost, if you will.
Okay, thank you. My final words are that, we're incredibly excited by what's going on in each of these technologies. We witnessed it yesterday with exon skipping and the evolution that's gone on in there. There's tremendous excitement and enthusiasm for what's happening right now in gene therapy as we learn about this. And as you get a sense of this technology, or these technologies that are coming in to address perceived weaknesses or gaps in the pipeline, and that's going to impact it over the next 3, 4, 5 years, you can get a sense of how all this is coming together. We haven't had a time, this hasn't been a long session where we're going to talk about combination therapies. I would have liked a little bit more time to get into that. But I think you can all get a sense today of what's happening now, the next couple of years, and then the next 3 or 4 years as all of these technologies come to bear in this disease. So with that, thank you, everybody. Appreciate it. Thanks, guys.